Hello everyone, welcome to back. Now let's start the chapter 2, okay? Let's talk about the tools of the laboratory. The tools of laboratory. Now in this section, we are going to learn the 5 I's and the different medias, okay? So what are the 5 I's? The 5 I's means inoculation, incubation, isolation, inspection, and identification. Okay. Now let's discuss those one by one. Inoculation means we have to pick the samples. Okay. So we have to pick the sample and let us pick the microorganism and let the microorganism grow in our media. So the medium. So plural is the media. So medium means the material provides the nutrients for the growth of microorganism. Inoculum means the small samples of the microbe. Okay. Inoculation means the introduction of the small piece of microbe into the material of the nutrients. That means into the medium. So we can get the piece. We can get the inoculum by scrub our mouse to get piece piece samples of microbe. In the in the hospital clinic, so the samples can be taken by the nurse with using the needles or the swab from the blood or the wound or the tissues. So once we introduce the inoculum to the medium, so we have to put the medium in a certain devices so that the devices can supply ideal conditions for the microorganism to grow. So the optimal condition is the temperature and the humidity. The temperature normally is from 20 degree to 35 degrees Celsius. So the incubator can adjust these temperatures. So we can also adjust the oxygen or carbon dioxide concentration in the atmospheric environment in the incubator to provide the optimal condition for the microorganism to grow. So, so the media can be put in either tube, glass tube, or the flask, like this. So the media is filled in the plastic field. Okay. So the media, we can adjust the nutrients in the medias to let the medias to let the medias to let the certain microorganism to grow better in the medium. For example, in this media, we can see those white colonies. This is the medium for the E. coli. Okay, so this is white colonies. Excuse me, these yellow colonies. These are the mediums for the growth of Micrococcus lutens. So red colonies. So this is medians for the growth of Serratia, Serratia macistrus. Serratia macistrus. So this is pure control. That means there is only one microorganism in the medium. We can have change, so we can change the ingredients in the medium to let two or more microorganisms to grow. So this is called a mixed culture. For example, in this flask, so this media can allow both E. coli and Micrococcus lutens to grow. So we can see these small yellow colonies. So these are the Micrococcus. So the white colonies, so this is the E. coli. Okay, E. coli. So this is E. coli, this is the Micrococcus. Also, so if the media is not specific, so when we culture a certain microorganism, for example, if we culture the Serratia macistrus, so these are the red colony. However, if the media is not specific for the Serratia, so the E. coli can also grow in this media. So this is the contaminated culture, okay? So that means, so this media is contaminated by the E. coli. Now the media is to supply the nutrients for the microorganism. So the media can be put in the glass tube, okay, flask or petri dishes, okay. So so we can introduce the microorganism by using the loops, okay. Loops is very thin metal wire, okay, or needles, okay, or the pipettes, or swabs, okay, swabs. However, all these tools have to be sterilized, okay, to avoid the contamination. So the media can be liquid or semi-solid, okay, or solid. So, for example, we widely use the agar. So agar is one type of polysaccharide isolated from algae. Okay, so when we so we can order agar powder from the companies. Okay, so we can mix the agar powder with the water and then heat the agar solution. So the agar can be dissolved in the water. So this is agar liquid. So we can heat at 100 degree, okay, and then we can cool down this liquid solution. Then when the temperature drop to 42 degrees, so the agar become a solid, okay, solid material. So this material is flexible and moldable, okay. So we can pour the agar liquid in the flask or the dishes. We can also mix the agar solution with all kinds of nutrients ingredients to make many different media for different microorganisms to grow. Here we can see, so this is liquid media, okay. So this is semi-solid. This is solid, like agar. So so we can classify the media to different categories. Okay, the first is synthetic, defined or synthetic media. So so we can order the chemicals from chemistry companies and use those chemicals to make the synthetic media. So this is very simple and basic media for certain microorganism to grow. Okay. The second is the complex media. However, for some microorganisms may require um, may require different condition to grow. Okay, so it's difficult for the scientists to culture certain microorganisms. For example, certain microorganisms require the extract of animals, such as extract of animals or plants or yeast to grow. Okay, for example, the blood, the serum, or the meat extract. Okay, milk, yeast extract, or either soybean. So we can add these ingredients to the basic synthetic media to make the complex media. Okay. So this is the formula of the synthetic media. We can see these are the amino acids. Okay, these are the amino acids. We can add also the sugar, the salts. Okay, vitamins. Okay, so these are all the chemicals we can order from the companies. So it's easy to make the media with these ingredients. This is the complex media. So we have to add the extract of brain, heart, okay, peptone, okay. So it's relatively difficult to get the brain or heart extract, okay. So I mean, so it's a little bit difficult to make this media. For example, this media is required for the Staphylococcus aureus. Based on the purpose, so the media can be classified to general purpose media. So general purpose media is used to grow a broad spectrum of microbe. Okay. Or the second is enriched media. Enriched media contains complex organic substance such as blood, serum, hemoglobin, or certain specific special growth factors. So some microorganisms require um, more nutrients to grow. So we had to use this unenriched media to grow those microorganisms. For example, so this is so these are two enriched media. So this is blood media, okay, blood media. So this is chocolate media. The chocolate media is also the media contains blood. So when we heat the media, so since the blood contains the iron, okay. So when we heat the blood, so the blood hemoglobin is denatured, so it changes the color, so the iron, so the compounds of iron change the color, so it looks like chocolate. Okay. So this is used for culture Neisseria. Okay, Neisseria. So we can see this white colonies actually are colonies of Neisseria bacteria. Selective media. Selective, we can add certain agents or chemicals to the media to inhibit or cure the unwanted microorganism, to let the microorganism that we want to get to grow. Okay, so it's very important in the preliminary selection of the microorganism from a sample that contains 10 different microbes. Okay, so we can add certain agents such as the soybean extract. Okay, soybean extract can inhibit certain microorganisms to grow. Okay, we can also add some antibiotics. Okay, to cure most unwanted microorganisms, and then to capture the microorganism that we want. Differential media. Differential media actually it can allow multiple microorganisms to grow. However, since the nutrients in the media will let the different microorganisms grow differently, so we can see so the colony size may be different. So the colony color may change. Okay, some colony may generate the gas bubbles. Okay, so if the microorganism have the capacity of the fermentation, so they can convert the sugar to the acid. Okay, to generate the gas, to generate the carbon dioxide. So we can see the gas bubbles around those colonies. Okay. So as we can see this diagram, so this is selective media. Okay. So if we add certain chemicals or reagents, so those reagents will kill the unwanted microorganism. So so finally, so we can get so we only get the microorganism, so the microorganism that we want to grow. Okay. However, the differential media, where since because we can see in this flux we have different microorganisms to grow. Okay, since we have the chemicals in this media, so some microorganism may get the purple color, some may get the red color. Okay, some may grow bigger, some grows smaller. Okay, so this is the differential media. So we can also combine selective and differentiated media. Okay, for example, McConkey agar can suppress the growth of some organism. Okay, however, it can so it's because the McConkey agar has the pH indicator, so it can also change colors. Okay, because the microorganism can convert the sugar to the acid, so it can change the color. Reducing media. So the reducing media. So for example, if the media, so if we add certain reducing agent to the media, so those reducing agent can absorb the oxygen. Okay, to decrease the redox reaction, to decrease oxidative reaction. Okay, and so So it can absorb the oxygen and decrease the penetration of oxygen to the media. So it can generate anaerobic environment in the bottom of the media. So it's good media to grow the anaerobic microorganism. And then we have the transport media. Transport media is the media to give to capture the microorganism to maintain and preserve the microorganism and for certain period of time, okay, for the transport. So here is the miscellaneous media, okay, miscellaneous media. And this media is to measure the fermentation function of the microorganism. Because it contains the pH indicator, it also, so this media is filled in a glass tube, okay. It also contains a small glass tube, so which is used as a trap for the gas, okay. So if the microorganism has the fermentation to convert the acid to convert the sugar to acid, so it can also release carbon dioxide. So this small glass vial can trap the carbon dioxide gas, so we can see the gas bubbles. So we can also see the change of the colors. That means so those microorganisms grows in this tube have the fermentation function. So we also have AC media. So AC media it will detect so it's used by the scientists for the drug test, okay. It's also used for the effectiveness of the disinfectant or the antiseptics, okay, cosmetic, okay. Enumeration media is widely used by the industry and scientists to count the numbers, okay, count numbers of the microorganisms. Now let's talk about the isolation, okay, isolation. Since the microorganisms have huge numbers and are ubiquitous, okay. So when we get the inoculum, okay, so the inoculum actually contains tons of microorganisms. So we have to isolate the microorganism that we want, okay. So we use the technique of the isolation to dilute and separate the microorganism. Once the microorganism is separated to individual sphere, so one individual sphere can grow to a discrete amount of spheres. So this is called colony, okay. So the colony can be visualized by our naked eye, okay. So this is the solid rising from the media, okay. So the isolation requires the medium, okay. So the media and dish, also the inoculating loop. So this is the isolation technique, okay, isolation technique. So this is inoculum. Inoculum contains all kinds of microbes, okay. So we have diluted the inoculum and separate them, okay, separate the different microbes. Okay, so once the microbes are separated to diluted and separated to individual cells, one individual cells can continue growth to the colony. Okay, to the colony. So we can perform this technique in the flask with the medium. Okay. So this is the method for the isolation technique. Okay. So we use the loop. Okay, isolation loop. So the loop is the metal wire. Okay. So first we have to sterilize the wire. So we can use the flame to burn this metal wire. So the burn can kill all the microorganisms in this wire. Okay, so the loop would be sterile. Then we can pour the inoculum in one part of the flask. Okay, in one part and then we scrub the surface of the medium with this wire. This way, okay, this way, like this, okay. That means we can bring a small amount of the inoculum and dilute, separate the microorganism in the inoculum, okay. And then we also pick some microorganism from the from diluted from the scratch. Okay, to do the scratch this way. Okay. So this performance continue dilute and separate the microorganism. Okay. So finally we can see. So the microorganism will be separated to individual cells. So individual cells will grow to the colony. Okay. We can also do the type B. We use the liquid media in the tube. So we use the loop to catch the inoculum and dip the wire into the liquid in the tube, and then dump the liquid media to the flask. And then we continue dip the wire to the second tube with the medium, and then we dump the media to the flask. Okay, and then we continue dip the wire to the third tube with the media, and then we dump the media to the flask with the solid medium. So as we can see, we continue dilute the microorganism in the liquid medium. So finally, so the microorganism in this liquid medium is individually, okay. So they will grow to individual colony, okay. So those two methods are used to dilute the inoculum with tons of microorganisms. If the inoculum contains only contains very few microorganisms, so we can directly pour. So we can directly put the inoculum in the liquid media and pour the liquid media to the solid media. Okay. So finally, because since the microorganisms are very few, so they are separated to individual in the liquid media. So once we put the liquid media in the solid media, so they finally goes to individual colony. Okay. Now let's talk about the inspection and identification. Okay, so we can identify the microbe with the microscope. Okay, we can also detect the cellular metabolism. Okay, for example, if the microorganism have the fermentation function, so they will generate the acid and carbon dioxide, right? We can also detect the identify the microorganism based on the products of the microorganism or the enzyme of the microorganism. So we can for the enzyme detection we can do the Western blot. Okay, since the enzyme is the protein, we can use Western blot. We can use Western blot to detect the specific proteins. Okay, we can also do the genetic and immunologic test. Genetic we can use the PCR. Okay, PCR. Immunologic we can use either ELISA or the Western blot to detect the proteins or antibiotics, proteins or antibodies. Okay. Now let's work on this question. Give an example of a specific type of media for each of the general category below. Okay. So I gave you guys 5 minutes, so you can review the slides and give me the correct answer. 1, 2, 3, 4, 5. Okay. Selective media such as the McConkey media, right? Reducing media contains the reducing reagents. Okay, so it can we can use reducing media for the anaerobic microorganism culture, right? So this is for the anaerobic. Okay, anaerobic. Complex media means we have to add the extract of the animals or plants or yeast, such as blood, heart, or brain extract. Okay, or brain. Okay. Differentiated media means differentiated media means this media contains certain reagents to show the size or color change of the colonies. Okay, for example, so it contains the pH indicator. Okay, so it allows several microorganisms to grow on these media, so we can see different colony size and colony colors. Okay. Now let's study the section 2. Okay. So we are going to look at the microscope. Now let's first start on the metric standard. Okay, metric standard. So because metric standard is the international standard. Okay, so we use this standard widely in hospitals, in labs. Okay. So we don't use the gallons, ounce, this standard. Okay. So the metric standard. So the basic length unit is the meter. Okay, it's meter. So we can So we have a gigameter. Okay, gigameter is 1 billion meter. Megameter is million, 1 million meter, and a kilometer is thousands of meter. Okay, thousand meters. Centimeter is hundreds meter. Okay, millions. Millimeter is thousands meter. We also have the micrometer. Okay, it's millions, and nanometer is billions. For example, the protozoa and algae are 3 to 4 milliliter. Okay, 3 to 4 milliliter. So we can see this microorganism. So we can visualize this microorganism with our naked eyes. Okay, the yeast are smaller. Okay, yeast are 3 to 4 micrometers. So we have to use microscope. Okay. So the virus are very tiny, so they are nanometers. So we have to use electron microscope. So here, as we can see this diagram, okay. So the loose and the mushroom, okay, so they are huge, okay. So we can see those living organism with our naked eyes, okay. However, for the protozoa and tissue cells or the bacteria, so their size are from 100 micrometer to 1 micrometer. So we have to use our light microscope to visualize those microorganisms. Okay. For the virus such as the HIV virus, okay, and the flagella, the DNA, so their size are from 200 nanometer to 1 nanometer. So for those microorganisms, we have to use the electron microscope. Okay, electron microscope. So the smallest one are the animal acids and atoms. Okay, so we have to use specific devices to visualize those particles. Now let's talk about the light microscope. So light microscopes are widely used in labs in hospitals. Okay, so it contains different parts. So the the top one is the ocular lens. Okay, ocular lens directly face to our eyeballs. Okay, and then so these are the objective lens. We have 4 objective lens. So those objective lens directly face to the slice, face to the face to objective, face to object. Okay. So this is arm. Okay, this is arm. So this is the stage. Stage is used to support the slice. Okay, slice are the glass slice that we can put the samples on it. Then this is the coarse adjustment knob. Okay, this is the fine adjust knob. So this is light bubble. Okay, light bubble can generate the light source. So let us to visualize the samples. Since the microscope can magnify the objective, can magnify the samples. So the objective lens provides the real image. So the ocular lens provides the virtual image. So the virtual image actually gave us magnification. Okay. So when we calculate the magnification, actually, both lengths contribute to the magnification. For example, if the objective length has 40 magnification and ocular lens has 10 magnification, so totally we have 400. Okay, so here as we can see this diagram, okay, so this is light bubble. Okay, this is light bubble. Light bubble generates light source. Light source can be reflected by the lens to the stage, to the stage, okay, through the slides, okay, and then so the image will be captured by the objective lens, okay. Then those images transferred to the ocular lens. The ocular lens is continuously magnified the image, and then so this image can be transferred to our eyes. Now let's talk about the resolution. Okay, resolution is also known as the resolving power. It is the capacity for devices to distinguish the distance between two adjacent objects. Okay. So if the devices can distinguish very small distance, that means these devices have the higher resolution. Okay. So our eyes have the resolution of 0.2 milliliter. Okay. So the microscope can reach to 0.2 micrometer. So high resolution means the image will be much clear. Okay, much clearer. For example, so this is high resolution. Okay, so the image is clearer than this one. Okay, so it's like a high. We have high definition TV, right? We can find the high resolution, high division TV. Okay, because high resolution TV can provide high quality of image. Okay, so we have four optical lenses. So the highest magnification is 1000. Okay, 1000. 1000 magnification. Actually, we can reach to 1000 magnification with 100 100 magnification objective lens. Okay, when we use 100 magnification objective lens, we have to use oil. Okay, so that's why 100 objective lens is called oil lens. Okay, oil lens is called oil lens. So why do we have to use oil? Because since this is a high magnification, that means this magnification need more light to go through. Okay. Also, since this objective is much more closer to the slides, that means we have to bend the lights to let more lights to go through to the objective lens. So if not, so the lights will be reflected out of the objective lens. Okay, so as we can see, so oil can bend the light beams, can allow more lights to go through the objective lens. So that's the purpose that we use oil. So the high resolution, that means it can distinguish distinguish more smaller distance between two objects, so it can give us a high quality image. Contrast. Contrast is also known as the refractive index. Okay, that is the degree of binding lights undergoes binding bias and pass from one media to another. Okay. So higher contrast means the sharper high contrast means the higher the difference in the refractive index, so the sharper the contrast. Okay, so that means it will give us more high quality of the image. Okay, so our eyes. So in the microscope, we have devices to adjust the contrast. This is called a condenser. Okay, condenser. We can adjust this and adjust the light beam to increase the contrast, to increase the image quality. Okay. We can also choose different microscope method to visualize the samples. One is the bright field. Okay, bright field allows all the light beam go through the objective go through the samples. Okay, so the background is white. Okay, so it's a good option to visualize the live samples, also the unstained materials. Okay, also the standard samples. Okay, so so the bright field is the most widely used method for the microscope. The second is the dark field. Dark field, so we can adjust the film to bend the light beam. To bend the out beam, to bend the light beam out of the samples, so only allow the light beam goes through the samples. Okay, so it can allow us to visualize the living cells more detailedly. Okay. So the phase contrast is another method. So we can adjust the light waves that passing through the samples. Okay, so it will make us to visualize the inner structure of the microorganism. Okay, such as the organelles. Okay, or the endospore. Okay, so we can use this method to visualize the inner structure of the microorganism. Fluorescence microscope. Fluorescence microscope. So we have to use ultraviolet radiation source to generate the fluorescence. Okay, so first we have to treat the samples. We have to stain the samples with the dyes. Okay, those dyes are the fluorescent dyes. Okay, so the fluorescent dyes can generate different colors such as green, red, blue. Okay, normally we have three fluorescent color, okay, green, red, and blue, okay. So once we stain the cells with those three fluorescent, okay, so we can shoot the samples with ultraviolet, okay. The ultraviolet can stimulate the fluorescence to generate a fluorescence, okay, and then we can use the microscope to visualize the picture, okay. So this since we have these three dyes, we can use these three dyes to stain the different molecules in the microorganism. For example, we can use red to stain the microtube. We can use the green to stain the proteins, to stain the cellular membrane. Okay, we can use blue to stain the nucleus. Okay, we can see. So the green actually they are the nucleus. Actually, so we can see the fancy picture of the cells. Okay. So all the fluorescence image actually, so they are black field. They are black field because we cannot use bright field. Bright field where attenuated fluorescence signals. Okay. We also have the confocal microscope. Okay. The reason that scientists invent the confocal is because if we use the high magnification to visualize the cells, since the cells are very thin, okay, so that means it's a very limited distance for the focusing, okay. So if we use the light microscope, so since this is high magnification, so we cannot visualize a slice perfectly. Okay, so we have to use confocal. Confocal use a laser beam. Okay, laser beam to scan the samples. Okay, since the laser beam is precise light. Okay, so it can get perfect the scanning size of the samples. Okay, so it can get the very so it can get the high quality image and very fancy image. Okay. So those what I talk about are the light microscope. Now let's talk about the electron microscope. Electron microscope is used to visualize very tiny microorganisms such as the virus. Okay, we have two types of electron microscope. One is transmission electron microscope. Transmission electron microscope uses the electron particle to replace the light beams. Okay, so since the microscope uses electron particles, so those electron particles have the very short wavelength. That means the microscope can shoot a very dense of electron particles. So it can generate a very high magnification. Okay, so the detector will receive the reflected or penetrated electrons particles and generated the pictures, as we can see here. Okay, So first we had to prepare the samples. The specimen had to be sliced very thin. Okay, had to be sliced very thin because this is high magnification. So we had to let the electron particle to shoot at all the to shoot at the equal size. Okay, so we can see this. So we can see very tiny. So this is very particle. We can see very tiny microorganism. We also have another type of electron microscope, scanning electron microscope. Scanning electron microscope also shoot the electron particle. Once the electron particle reach to the samples, it will be reflected and detected by the and received by the detector. So that's why so scanning electron microscope can be used to visualize the surface of the samples. Okay, since since it requires the reflection of the electron, so we had to prepare the samples with a specific method, such as coating the surface of the samples with the metal. Okay, metal coating. Okay, normally we use the very expensive metal, such as gold, to coat the samples. Okay, so we can see this is a picture of scanning electron microscope. Okay, it's widely used to visualize the surface of the samples. Now let's talk about the preparing samples. Okay, preparing samples. So the specimen had to be prepared for the microscope to visualize. Okay. So the preparation can be simple or complicated. Okay. For the living or died, so based on the observations, we can use the living microorganism or died microorganism. Okay, we can choose the different methods such as bright field, dark field, or phase contrast. Okay. For the living preparation, so actually it's very simple. Okay, since we had to keep the microorganism live, so we don't have to do more treatments. Okay, so maybe we just use a drop of the cultural media that contains the microorganism, and when we drop the media to the a slide, so this is called a white mount. Okay, so we can so we can observe the specimen with the microscope. We also have the high drop. Okay, high drop. So we put the media in the concave sides and sealed with Vaseline and covered with a tape. Okay, like this. Stain. So we can use the stain to color the different structure of the microorganism to let us better visualize the microorganism or for identification. Okay, so we can use the dye to stain. Okay, we have two type of dye: basic dye and acid dye. Basic dye have a positive charge. Acidic dye has a negative charge. Okay, so the bacteria normally will attract the attract the basic dye. Okay, because the bacteria are negative charged. We also have negative or positive stain. Okay, so this is not a positive or negative charge. Okay, positive stain means the dye stick to the specimen and gives color. Okay, negative stain means doesn't stick it to the specimen and doesn't color the specimen. Okay. For example, negative charged cell will repair the negatively charged dye. Okay, so the cell will not be stained. Okay. Sample stain means we just use one single dye and we just use one single dye and we can stain the cells so we can get a perfect image. Okay. Differential stain, that means we have to use two or more dyes to color the samples. Okay. For certain microorganisms, we have to use these differential stains. So we talk about this later on. So the simple stain like a crystal violet stain on the E. coli. Okay, so just one dye. So one dye can color the cells. Differential dyes differential stain. For example, the green stain. Okay, green stain is developed by the Hans grains. Okay, so it consists of four steps. One is the crystal violet. Okay, so this is primary stain. Okay, another is the Grans iodine. Okay. And then we have to use alcohol to rinse to decolorize. Okay, and then we use the counter stain. We use the saffron. Okay. Since the differential stain can differentiate the microorganism. So if the certain microorganism is gram positive staining, so they will be differentiated from a gram negative staining microorganism. Okay, so we can use this staining to differentiate the microorganism. So it's widely used to the bacteria classification and identification. Okay. The second differential stain is acid-fast stain. So this type of stain is specifically used for the tuberculosis or leprosy or leprosy identification, because those bacteria cell walls contains the waxes. Okay, so the waxes is one type of lipid, so it can make the bacteria's cell wall waterproof. Okay, so it's difficult for this bacteria to be stained with either simple stain or the gram stain. Okay, so those stain cannot be used to stain these bacteria such as Mycobacterium tuberculosis or the leprosy. Okay, so we have to use acid-fast staining. Because when we use this stain, okay, so we will use acid alcohol to decolorize. Okay, since this bacteria's cell wall contains waxes, so the waxes can hold the dyes in the cells. Okay, so the cells still have the color. Okay, compared to the other cells. Okay. So this acid fasting is also important to identify the fungus protozoa. Okay. So the third differential stain is the indoor spore stain. It's similar to the acid-fast stain, however, it's forced by the heat, okay, into the resistant body, because the endosporal is a very tight tissues to let the microscope resistant to the extreme conditions. So we can use this endosporal stain to identify the microorganisms such as bacillus and clostridium. Okay. So here is the gram stain. Okay, gram stain. So the blue is positive. Okay, red is negative. Okay. So this is acid fast stain. So we can see those as the tuberculosis. Okay, tuberculosis. This is the endosporal stain. Okay. Now let's talk about the spatial stain. Okay, spatial stain, for example, for the capsule stain. Capsule stain, actually, we use the negative stain with the India ink. Okay, India ink. Actually, we use this ink to stain the background. Okay, not stain the microorganism. Because the capsule is also a tight, very tight material, so it cannot allow the stain penetrate to the microorganism. So it's difficult to stain the microorganism. However, since this microorganism has very thick capsule, so we don't have to stain this microorganism. We just stain the background and let the light beam go through the samples, so we can visualize the so we can use capsule. Okay. The second is flagella staining. Okay, flagella staining. So the flagella actually is the tiny filaments that is used by the bacteria for the locomotion. Okay, locomotion. So if the bacteria have the flagella, so we can stain the flagella for the identification. Okay. So here we can see this is the capsule stain. Okay, capsule stain for the Cryptococcus neoformans. Okay. So this microorganism has thick capsule. Okay, thick. So this is thick capsule. So this is flagella. We use the flagella to stain the flagella stain to stain the flagella. If we find the bacteria has the flagella, so that means this different from the other. So this microorganisms are different from others. Okay, for example, this stain is used for the Proteus virus. Now let's look at this question. Okay, identify each of the staining techniques as sample, different, and special. So the differential staining are the. So I give you guys 5 minutes, 5 seconds. Okay, 1, 2, 3, 4, 5. So differential staining are the green staining. Okay, green staining. Acid fast staining. Okay. So the special staining actually is the capsule staining, right? Capsule staining, and the flagella staining, right? Flagella staining. So the crystal violet actually is the sample staining, right? Now let's move on. Now this is the appendix of all the containers that I have covered in this chapter. Okay, so you guys feel free to review all the contents with this appendix. Okay, so this is the end. Okay, bye.